This lecture is going to take you a bit more in-depth into protein synthesis. So in the last lecture, I talked generally about transcription and translation, both of these being involved in producing proteins. Remember that your cells are basically mini protein factories and that you are in fact composed mostly of protein and water. And so these proteins are getting produced every second of every day by almost every cell in your body. Those proteins need to be produced correctly, otherwise you can have things like disease. And there are multiple types of RNA involved in the process. You get a little hint of that because here this says mRNA, and so far we've just been using the general term RNA. So we're going to talk about all three types of RNA and then go into protein synthesis a bit more. This is table 13.1 from your book. What it does is it shows you the three types of RNA, and all three of these have different functions. And remember that in biology, form is function, so they're going to look different, and that's going to dictate how they act. Now, we've been talking about RNA so far basically as a copy of the gene, right? So, copy of the gene with regard to protein synthesis. And so that would be, in fact, mRNA. All right, so we've basically been talking about mRNA. So this copy of the gene is what is made in the nucleus, leaves the nucleus, and it's what you saw in this image. That's this guy right here, kind of this ribbon shape. So made here, just a copy of the gene sent out to the ribosome and is read. But there are other types of RNA too. There is ribosomal RNA, and there's, very importantly, transfer RNA, tRNA. So we're going to be talking about mRNA and tRNA mostly. Ribosomal RNA is actually incorporated into the ribosome. There is some code on it, and so it is an RNA molecule. It uses U's, but that's going to be incorporated into the ribosome. We're going to be working with tRNA and mRNA. So if mRNA is a copy of the gene, and its function truly is to copy the code of the gene, to create an exact copy so that that can be spelled out on the ribosome and the amino acids can be linked in the correct order, right? So this is the information coding molecule. Then the function of tRNA is to bring the correct amino acid. So this molecule is a hybrid molecule. You've got a protein in the middle, and you have an amino acid on one end, and you have code. Notice that this is in a group of three, so 1, 2, 3. And that RNA code is called anticodon. And that's because your mRNA is broken into groups of 3, and I'll show you that in an actual code in just a minute. And each of those sets of 3 is called a codon. So mRNA has codon code and tRNA has anticodon code, and those are actually going to be matching one another. Okay, which I'll show you in the next slide. Now, what's interesting about tRNA, it's this hybrid molecule. This amino acid is actually specific to this anticodon sequence. So whatever the sequence is here, it will always only bring that amino acid that's associated with that sequence. So what I'm telling you here, the big picture, is that this process is extremely precise. You have the code that spells out which amino acids go where, and you have tRNA that brings the exact amino acid that's needed. This is extremely precise. It's originally coming from DNA. DNA. That gets copied into this long list of codons, and then tRNA brings in the appropriate amino acid. So let's take a look at how this works. Let's draw in a ribosome right here. It's got kind of this smallish subunit and a larger subunit, and what happens, let's label it first, ribosome. what happens is mRNA is read through it, and it's read in codons. So there's A, G, C here. And as that gets pushed through the ribosome, that codon is read, and tRNA goes ahead and it base pairs. So here you can see this is correct RNA to RNA base pairing, U to A, C to G, G to C. And because that says UCG, then it brings only serine. This protein part is just sort of the mass of this macromolecule. It's just holding the two important ends, one being the amino acid and the other being the code. So as this mRNA is fed through, you have multiple tRNAs that are going ahead and bringing in amino acids. Okay. So one of the things we're going to want to do is look at the amino acids. So here's an amino acid table, and it uses codons. So it's actually called a codon table, and you use codons to look up particular amino acids. So let's go back, and if we look here, we're going to be looking at codons to look up what amino acid it is. So A, G, C, right? And we see that that brings serine. So let's go take a look on here. So AGC, right? So how you use this is you use mRNA. You don't use tRNA. You want codons. So you've got A, you've got G, and you've got C. And so you just kind of come over A, G, C. AGC, there it is, and serine it is. Now, I do want you to note that there is redundancy built into this table, and that's really interesting. Some have more redundancy than others. So notice glycine. You have GGU, GGC, GGA, or GGG. So all of those code for the same amino acid. And it's thought that this redundancy helps with combating mutation. Remember, eukaryotic cells conserve their DNA. So let's say that the DNA was transferred into mRNA, and the mRNA strand said GGU, and that's what it was supposed to be, and it's supposed to bring glycine. Well, let's say there's a mutation in DNA that leads to an incorrect copy of mRNA, and let's say that mutation led to a GGC in the codon. It wouldn't matter. You would still get glycine. So this is amino acid redundancy, and so you can have mutations that lead to a change in the third letter, but you can still get the amino acid for some of these mutations that might occur. So remember, mutation is always in the forefront of our mind when we're thinking about this process of protein synthesis, because what we want to know is, gosh, well, what happens when things go wrong? And when you have mutations in DNA, that gets copied into mRNA, and then that gets translated out on the ribosome, so that, you know, instead of the square, you end up with something like a triangle, and that can lead to disease. So things like sickle cell disease and cystic fibrosis, which we'll be able to talk about in class. So one of the ways to combat this is amino acid redundancy. So amino acid redundancy. The other safeguard we have against mutation is proofreading. And what that is if you have a mutation in DNA, so this little black dot, what you have on your DNA are proteins that hop on the track and check DNA. So they look for conformational changes in the strand, and then they will excise the mutation and bring in the correct base pair. So you can actually correct DNA so that you don't get the error in mRNA, and then of course the trickle-down effect of the messed up protein potentially. So proofreading is another way to combat against mutation. The third way to combat mutation would actually be cell death. And so cell death can occur if you have DNA that is mutated and you're constantly producing this incorrect protein. If DNA is mutated too much, then this cell can program cell death. and avoid producing mutated proteins. Okay, well, that concludes your talk on protein synthesis. A little bit more in depth with this process. And what we'll do when we're in class is we'll look at video on this so you can actually see this in motion.